This is a summary lesson on transpiration, and you can see the plan over here. So I'd like to just go through an overview on transpiration. What does it mean? Why do plants require it? We need to know where the water's leaving, and that's why I'm moving on to guard cells afterwards. Just a very brief comment on that. We then want to touch upon the different types of potometers you could come across with some exam questions, and a brief comment on why plants need fresh water. What's the purpose of it, and some reasons for that. And then finally, where does the water go for photosynthesis? The types of potometers and common sort of factors that will increase and decrease transpiration comes in here as well, and I'm going to move on to that in due course. So water, provided there's a good concentration in the soil, will enter in via these roots into root hairs, and it will then start to move into these tubes called xylem. Now xylem are these very thin tubes that run right through the centre of the plant, and I've just drawn one illustration here, and the water will go up through that central tube and come into the leaves. It's going to go into a series of cells, and then eventually, once it's been used for the processes of photosynthesis, starts to form all of this water vapour, which you can see. And it then exits via these gaps in the bottom surface, mostly of the leaf, which are called the stomata. What surrounds these stomata? Well, that's the picture that we can see down here, which are the guard cells. Now, I've shown you two examples of a guard cell, and this one is a full of water guard cell, and you notice that the inside of the cell has got much more cell wall, and that allows the cell to take this kidney bean shape, which means that when it's full of water like this, the hole or the gap in the middle, which is called the stomata for many of them, or the stoma for one, is open. Notice when a plant isn't full of water, the stoma is closed, and that's going to be a big problem, not only for water loss, because water is going to be coming out of this, but also don't forget carbon dioxide. Carbon dioxide is going to struggle to get into these pores if it's closed. So let's now move on to some potometers and have a look at a few different examples. They all have the same principle, and that is that this principle that the water is going to be going up through this plant and exiting out via the undersurface of these leaves here. And they're all different design, but they show the same principle, that if you have a stopwatch and you time it, and in this example here we've got a syringe which we can plunge down, and that's going to get our water level up into this capillary tube here, and then over time, as the water comes up and out from the stem and out through the leaves, this capillary tube line is going to go down, and you can measure that in time and the amount of distance that's been lost in that capillary tube, and all you would need beside it is a little ruler to measure the sort of distance. And if you wanted to be even more technical with the maths, you could actually measure the volume, so long as you knew the diameter of the capillary tube. So that's our first potometer. The second one is the one that we covered in class, and it's a similar design, but we've now got an air bubble, and this has been injected into the capillary tube, and the rest of it is all water. So this is a solid column of water running right through this tube, which I'm just putting in just now, minus the bubble that you see that's going to sit at one end of the capillary tube. You can see the ruler that's below, and again we can measure the distance that this air bubble is going to travel, and clearly that bubble is going to move this way as the water is leaving the plant leaves via the stomata here, and the distance that it travels is obviously can be worked out as the rate of transpiration, which is again why we've got a stopwatch. The final potometer is a mass potometer, and if you think about this for a minute, we've got a mass here at the start, which you can see here is 234.62 grams, but as the plant loses water from the container, so it's coming up through here and coming out and leaving as water vapour, then clearly the mass of the plant is going to go down over time. And again, we can time that and calculate a rate of water loss. So that gives you a good indication of what these potometers can look like and how to understand each different version. So why do plants need fresh water? Well, with fresh water, you can see the guard cells are nice and open. Sorry, the guard cells are turgid, they're full of water, and the stomata, or the stoma in here, is open. And that means that we can get CO2 coming into this plant, which is an essential ingredient for photosynthesis. Because all the cells are full of water, the leaves are up and out, and they are competing for light energy up here, and this plant is competitive, so it's doing its job and it's being successful. Conversely, with no fresh water, the guard cells are going to be flaccid, so the stoma is going to be closed. It's going to struggle to get CO2 coming into it, hence why I've illustrated no CO2 here. The leaves are wilting, the plant is wilting, so the leaves are not competitive for light, and it's really going to struggle with that. Furthermore, every time we get fresh water and minerals coming into these plants, well, it's bringing in mineral ions as well. And we've done some work on nitrates, and you know how important that is for making DNA, amino acids and proteins. So fresh water means fresh mineral ions as well. And as I've said, nitrates are an important component for all plant and algae and the work that we did with eutrophication. So we can see the importance of that. And finally, on a hot day, it does actually help to cool the plant, because the loss of water vapour out through the stomata does take heat energy away. So far, we've touched upon the following areas. We've done an overview of transpiration, where this water is moving from root hairs up through xylem and out through the leaves, evaporating as water vapour. We've looked at the guard cells, and we can see how they want to be full of water to open up the stomata. And we've seen the different types of potometer, a mass potometer, and ones where you can see the water level changing in a capillary tube. We've also talked about why plants need fresh water: to bring in fresh mineral ions, to keep the cells full of water and turgid, which allows the leaves to be upright and out, and competing for light, and also getting in carbon dioxide. So we're now going to have a look at where the water goes for photosynthesis before we go on to looking at the sort of reasons why transpiration will change with different factors. As part of your course, you're supposed to know about the structure of some of the cells inside a leaf. And what have I done here? Well, I've taken a leaf, a blade of a leaf, which is this bit here. You don't need to know that, the leaf stalk or the leaf petiole here, but I've drawn a very abbreviated version of some of the xylem here, and we can see the water going through it. I've then taken a cross section of this, and you can see I've cut this vein just in the centre here. And this is the cross section image over here. So this is my cross section that we can see that I've taken as a drawing on the picture to the left. We can see the left and right side of the leaf. This is the left side over here, and this is the right side over here. And we can now see that we've cut the vein, which is this leaf vein here, and we can see the xylem that's sitting at the top of the vein up here. The sugar-containing vessels are down at the below. What I've then done is I've then taken a cross-section of this piece of the leaf. So we've got the top surface, which we can see here, of the leaf at the top here, and we've got the bottom surface down here. And I'm going to magnify that up to show you what this image looks like. And here we are. So this is what we see when we're looking at that top bit of the leaf. We've got first of all, our waxy cuticle, which is the bit that just gives the leaf the waxy sort of appearance on the top. We've then got some cells that don't really do photosynthesis, but they are cells, but they let light to come through to the main photosynthesizing cells below. These are the upper epidermis cells, and as I say, they are generally transparent, and they allow light to go through into the main photosynthesizing cells, which we can see, which are these palisade cells. All right, these are the palisade cells here, and you can see how many chloroplasts there are. There's perhaps as many as 300 or 400 chloroplasts inside each of these cells. So when the light is passing down from above, then the light energy is really wanting to sort of start to hit these chloroplasts in these palisade cells. So the cuticle needs to be see-through, which it is, and the upper epidermis needs to be as well. Notice I've drawn some blue arrows over here, and that's because the xylem and the water from the xylem is going to be coming over and providing the water for photo. We've got this water appearing here, and that's going to be going into the chloroplasts, and it's going to be bathing those in water so that photosynthesis can take place. So all of this is going to fill, just illustrate one with you here, it's all going to fill up with water that's going to help this cell to do photosynthesis. We've then got an important area in terms of transpiration, which is called the spongy mesophyll area, and I've drawn that with you quite carefully. And you notice that it's spongy because there's loads of gaps in between the cells. And those gaps really help the water vapour, any that's not used by the cell for photosynthesis, to gather in between the spaces. And that's why I'm just filling these in now. So we've got loads of water vapour coming into these areas. And then finally, if the water vapour is escaping, which we've got an illustration of that through these holes here, they're escaping through the stomata. And you can see that there are cells either side of the stomata, and these are the guard cells. So I've drawn one of the two guard cells there, and this is the stomata here. So this is the gap in between the two guard cells. Notice that I've drawn the water vapour, and that's evaporating. Notice that the key word there is evaporation. So that's how the water vapour escapes from the bottom of the stomata. So we can now see how important the water from the xylem coming into these cells is, and also how the water vapour can escape from the bottom of the leaf, which is what we look at when we look at transpiration. So now moving on to the final bit of the lesson here, transpiration. What can we see and what do we need to know about it? Well, we've done some work in lessons, but here's a little bit of content if you weren't here. I've gone through some of the factors that increase transpiration in the first instance, and we can see that we've got the example of wind. We're going to look at that in more detail in a minute. I've gone through the example of temperature over here, and we can also mention the number of stomata that you've got, which will directly impact the amount of transpiration that you're going to have. So let's talk about wind first of all. So what I've got here is I've obviously got an illustration, but I've also got a description. When it's windy, you're going to blow the water vapour directly away from the stomata. And by doing that, you're going to create a much greater opportunity for the vapour that's inside the leaf to leave and exit. So we can see here that the wind has blown all of this stomatal water vapour that was up here in the entrance of these stomata here. It's blown it straight away. And so what that's doing is it's encouraging more water vapour to come out. And that's going to increase your transpiration because you're creating a bigger concentration gap between inside and outside of the stomata. Temperature, well, we can see temperature, we're going to increase the movement of water molecules. And for those of you that do physics and are happy with physics, this is increasing the kinetic energy. So when you increase the temperature, you're increasing the kinetic energy. And we can see that with this illustration over here on the right. We've got our water vapour that's sitting in the inside of our leaf here, but we've got now an awful lot of movement, so much so that actually a lot of the water vapour is going to be moving out of the stomata, because of the fact that we've got much more kinetic energy, much more movement energy in the molecules, and again, that's going to increase our transpiration. Finally, the number of stomata, clearly the more you have on a leaf, then the more water you're going to lose. And the two illustrations here, this leaf here, only 3 stomata drawn, and some water vapour is leaving. However, if we look in exactly the same conditions, but with a leaf with many more stomata, well, you can see for yourself that there's so many more opportunities for the water vapour to exit from that plant, and that's clearly going to increase the rate at which it's going to lose the water vapour. The illustration in our temperature and wind as a sort of laboratory was, well, when we did our temperature, we looked at a hairdryer with the heat setting on, and wind, we just looked at the hairdryer without the heat setting on. Decreasing transpiration then. So there are things that will decrease transpiration. Humidity is the number one thing really to talk about, but sometimes you will get a question alluding to what might happen to a plant if you block the stomata. But we'll do humidity first of all. Remember, humidity is the concentration of water vapour in the atmosphere. And so if we increase the concentration of water vapour in the atmosphere, and we can see we've got a lot of water vapour around the outside of this leaf in this diagram here, we're going to really struggle to get that water vapour from inside the stomata out into the atmosphere. The example that we gave in the class was by putting a plastic bag over this plant. That's going to, if you leave it for about an hour, increase the humidity. And increasing the humidity from the initial water loss that's happening from the plant. Remember, this plant all the time is getting water in through the roots, up through the stem, and then out through the leaves. So it's going to be increasing, so long as you seal it up, the humidity levels to a point when, at that point there, you're virtually going to get zero transpiration afterwards. The other example, which we've talked about, is blocking the stomata. If you block the underside of the stomata, and in this example here I've chosen some Vaseline just to block it. Vaseline is this petroleum jelly stuff that is oil based and an excellent sealant, and it will block off the stomata on the underside. Clearly, as a result of that, you're going to get no transpiration coming out from there. So finally, what should you take away from this in terms of the lesson? Well, as much as the content, which has been important and is significant to your course, is this ultimate understanding that if you've got a large concentration on one side and a small concentration on the other side, if you can establish that, which is basically what we call a large concentration gradient, then you're going to get a lot of water loss going on, and that's going to be high transpiration. If you've got a low or a short concentration gradient, or a shallow or not much difference in concentration between one side and the other, and here is that example, and remember this is when we talk about the condition of humidity, you're going to get very, very little water loss, because transpiration, water vapour, will not move from this location which is high to this location which is also high. This one, however, will. It will go from a high concentration here to a low concentration, and it will go quite quickly. So what have we done in terms of the lesson? Well, I think you would agree that we've now covered where the water goes for photosynthesis, and we've also touched upon the factors that are going to affect transpiration. And so you now should have a decent understanding of all of those components that you're going to be perhaps asked questions on in your future exams. Hope that was useful for you. Synthesis.